Here we go, game on. Oh, you guys were quick on that one. Red blood cells carry. If you're stumped at the beginning, you might look at the pictures. They should be helpful to you. Function of hemoglobin. We maybe didn't talk about ribosomes, but you should have remembered that. Woohoo, nice work, kiddos. Erythro means red, thromb means getting their point. This is a tough one. So most of you are going to say spleen because that, oh no. It was in the reading, it's in the picture, and you might just go with context. Erythro meaning red, so $120 days, almost all of it. Nice work. Kayla's on fire. I meant mom. You guys are rocking it. Transport of respiratory gases. Ah. 'Cellular components, not chemical components. The green where press the formed elements is the cellular components. When you give blood, you're going to give whole blood, plasma plus cell. So next thing I wanted to do was, so getting into that routine of little things at the beginning of the hour, then our content, and then little things at the end. Hematopoiesis, biozone, that's in today's posting, today's Google Classroom. Hematopoiesis, biozone. Wrong, sorry. Blah, blah. Okay. It should be a Word document. So I like these. These are like single pages, kind of like your Pogos, but short. You got a little bit of reading, context. Pictures help you out. Another little bit of reading. If we were in class, we would talk to the text. Well, I'll pull that up later. I got too many other things open right now. If we were in class, we would talk to the text and then answer the 5 questions that follow. So I'm going to give you about 5 minutes, and you'll probably be into the questions at that point. Okay? So if we were in class, we would typically talk to the text over this. If you guys, it's been a while since you've talked to a text, maybe you might forget. Some of you highlight, and I noticed some of you did that with your highlighter on your document, or maybe you underlined, and you can do that still with documents. When I do that, I also take notes. I kind of like read and then I make sense of it, and so like this is my own sense making a flow chart of how words were connected, identifying some key terms. Right here, it talks about what's unique to stem cells. It was interesting that each cell type has a different growth factor, which makes sense because your DNA codes for these proteins or these genes, right? So the cells are made according to what genes are expressed. So it would make sense that they each have their own growth factor initiating it. I talked about these different cells and what they do, so I identified their functions as I was going. I tried to make sense of the picture as I go. Okay, so typically we would do that. So you were to, I would call on you, but I have the answer key right here, so I might as well just show you. Describe 2 defining features of stem cells, so what makes them different or unique than other cells, which I found right here. Can differentiate into other cell types, and they're self-renewing, so they can make more of themselves. Identify the blood cell types arising from each progenitor. So last hour, a lot of kids got this wrong. They were giving me just lymphoid progenitor, or lymphoid, or mastoid. I'm sorry, myeloid. But what it's really asking is what comes next. So the lymphoid precursor comes before. The lymphoid stem cell makes T cells, B cells, and natural killer cells. The myeloid precursor makes your granulocytes, neutrophils, basophils, eosinophils, red cells, and platelets. So that's what it was asking. What cells do these stem cells make? Number 3, use an example to explain the purpose of stem cells in adults. So stem cells are going to make more tissue, right? So to maintain and repair tissue, if you have a tear in a muscle, for example, it's got to make more muscle to fill that tear. Give rise to blood cells. We know our red blood cells only last 120 days. So obviously we're going to need to make more red blood cells. Bone cells, you get a break, cartilage cells, neuron cells, making new tissue of any type. Other places you can find the stem cells other than red bone marrow, and I didn't really see this in the reading, or at least I'm not finding it right now, but skin, brain, and digestive tract, you have stem cells in all those areas. And then why would you need new blood cells to be created? They have a very short lifespan, right? 120 days, a week, depends on the type of cell, hours. So we're going to need to constantly make more blood cells. So the making of blood cells is hematopoiesis. Okay, so that's today's topic. This was a confusing one to read about, so we want to go into more detail. Do you need more time on this to check your answers? Or am I good to go? Thank you, I appreciate that. Okay, so stick around later if you want to go over that again. On our agenda, we're going to do hematopoiesis, and what we're going to do with our notebooks is update them with 6 more slides. So you turned in 6 slides to begin with. We're going to add 6 more. And then flex time, you're going to read about hemostasis. So that same 3 levels reading guide, spit out, regurgitate, interpret. So let's look at this hematopoiesis. So first, what I want you to do in your Google World, open up the notebook posted today. You do not have your own version of this, but this is a growing notebook, okay? So what we're going to do is we're going to take 6 slides from this and we're going to copy them into our other notebook. So starting on slide 7, we're going to copy through slide 12. So I use Control and Shift. That allows me to select multiple slides at the same time. So I click on 7, Control Shift, click 12. Not sure if you have different controls. Okay, so now I can Control V or C for copy. I can go Edit, Copy, or I can right click and copy. So Control C, Edit, Copy. Then go to your notebook that you did last week. So week 20, you posted it. You actually have this in your files. You don't necessarily have to track it down this way. Week 20, open up the notebook. So I'm going to open up Honesties, and I'm going to go to slide 6. So in between 6 and 7, I'm going to click, and then I can paste. Either Control V, Edit, Paste, or right click. It's going to ask you to match styles in this or keep original. Select Keep Original. so that it doesn't change the template. So now you have 6 more slides that you can work with. So keeping one notebook for this chapter. This is going to be your note-taking slide. So I'm going to talk about these 4 different processes. I don't have a lot to say. We watched some videos. Each slide that follows is going to be like the follow-up to that process. So when we do erythropoiesis, we're going to watch a video, and there's 1 slide to, you got 3 squares to do. When we do leukopoiesis, go to the next slide. Here's questions to fill out during the video and a follow-up. Then we're going to do thrombopoiesis, then the 3rd slide, questions for the video, and this is just a reference sheet, okay? So we'll fill those 9, 10, 11 out when we watch our videos, and then the follow-up questions like we did last time, okay? Is anybody struggling to get the pages copied onto their notebook? Okay. If nobody is telling me they need help, I'm going to start the PowerPoint. So first of all, that square about hematopoiesis, you definitely want to know that it is blood cell formation, the process of making new blood cells. So AKA blood cell formation, anything like that. I should put this in presentation mode so it's bigger. For each of these, we're going to want to know what cells give rise to them. We're going to want to know the chemical that stimulates it. So you read a little bit about stem cells. You identified some uniqueness in that they're undifferentiated and they can become any type of cell. When you were conceived, that zygote, egg and sperm come together, that zygote is a totipotent stem cell, meaning it has total potential. So it can become any type of cell, any kind of cell. And then it starts to divide and it'll become a pluripotent stem cell. A pluripotent stem cell can become one of many types of tissues, but not all tissues. So you first have 4 types of pluripotent stem cells. And then those stem cells continue to differentiate, right? So you have what's called the hemocytoblast. Here's that word down at the bottom, hemocytoblast. This is your stem cell that gives rise to all types of blood cells. This happens in the bone marrow. So where? Bone marrow. Or you can call that myeloid tissue. So marrow, myeloid, M and M. We want to know this word hematocytoblast. It's your main blood cell stem cell. It becomes any type of stem cell. It will first differentiate, so lineage, into a myeloid or lymphoid. You do want to know those 2 words. Myeloid stem cells are found in the myeloid tissue, bone marrow. Lymphoid stem cells give rise to cells that will leave and go into the lymph tissue. So lymph vessels are like blood vessels, only they contain plasma. Okay, so here's a flow chart. So here's your multipotent stem cell, your hematocytoblast. Hemo, blood, cyto, cell, blast former, right? So it's the one that gives rise to all cells. Then it's going to make a secondary stem cell. The 2 with the little circle at the top that I used, that's secondary. So your secondary stem cell is either lymphoid or myeloid. You call them progenitors because they're pro-generation. They come before. They give rise to precursors. Like they're all different ways of saying the same thing. So the lymphoid, we want to know what cells they form. So they're going to form B cells, T cells, and natural killer cells. End result. Myeloid forms, end result here, all our granulocytes, thrombocytes, platelets, and monocytes. Okay. And you kind of answered that question on your biozone already. It's kind of interesting, later on I'm going to refer to the hormones erythropoietin and thrombopoietin, so very similar words. So notice they're coming from the same precursor, the megakaryocyte. So it's kind of makes sense that they would have a similar hormone that initiates that process. Okay, you should be able to fill out a chart like this. So you saw this, the hemocytoblast, your primary stem cell, the lymphoid, secondary stem cell that stays in the, produces lymphocytes, B cells, T cells, natural killer cells. The myeloid is marrow. These cells will stay in the marrow. So all your granulocytes, all those that end in phils, we can recognize them by the shape of their nucleus. The monocyte, the giant one, platelets, and erythrocytes. Okay, so practice making that diagram. You did this in the reading assignment last week, and a number of us, they did a different flow chart, erythropoiesis rather than hematopoiesis. So hematopoiesis, all cells, whereas erythropoiesis, just red cells. Okay. So erythropoiesis. This is a good flow chart here. You can see that when the kidneys, that's this right here, the kidneys, they sense hypoxia, hypo, low, oxy, oxygen. So when you have low levels of oxygen, which might be because you're just going through oxygen really quick, like exercise, you're at high altitudes where the air is thin, you have anemia, maybe not enough cells, you're low on iron. Any of those will cause you to have low oxygen levels. That stimulates kidneys to release erythropoietin. Erythropoietin is a hormone. It travels through the bloodstream, tells the bone marrow to stimulate red blood cell production. So you want to know the hormone, erythropoietin. You want to know the stimulus, low oxygen. And you want to know the kidney produces the erythropoietin. So when oxygen levels are low, kidneys release erythropoietin, which stimulates red blood cell formation. On each of these slides, I'm going to watch a little video because I like you to be able to visualize the process. So they're all relatively short, like a minute, two maybe.
Erythropoiesis is the process of erythrocyte development. It begins in the bone marrow when pluripotent stem cells differentiate into erythrocyte-specific stem cells. These cells generate colonies of proteurethroblast that, in response to erythropoietin, further differentiate. The erythroblasts form an island around a central macrophage that nourishes the cells. The basophilic erythroblast begins to produce hemoglobin, changing the cell into a polychromatophilic and then orthochromic erythroblast. Before leaving the bone marrow, the cells cast their nuclei to the macrophage and enter the bloodstream as reticulocytes. The remaining organelles degrade and the cells become mature biconjugate erythrocytes.
So that just gives you an idea of how the cell has to change, and a certain chemical will tell it what changes to make, right? Because remember that hemocytoblast can become any type of blood cell. Leukopoiesis is the formation of leukocytes, right? The formation of white blood cells. So remember, we identify them based on their nucleus. The multi-lobed neutrophil, the headphone-looking nucleus, the eosinophil, the basophil that has really large granules, so you hardly see the nucleus. Your B cells and T cells, those lymphocytes, kind of look like eyeballs. The monocyte has that kidney-shaped nucleus. So there's two chemicals, interleukins and colony-stimulating factors. So these are proteins that would turn on genes in that stem cell that tells it to become the different types of white blood cells. So depending on the type of infection, our body will send signals that we need certain types of white blood cells, right? So it'll release a different interleukin or colony stimulating factor to turn on that particular gene. So we'll watch a video of this and you'll see how that nucleus changes shape as it begins to differentiate. And you can see at one point then there's no turning back, right? Because the nucleus is all different shapes and you wouldn't be able to cut it in half. You'll also see in the end how the different chemicals they carry, like our basophil carries the serotonin and histamine. So you'll see those chemicals start to be formed, and it's all part of that cell differentiation that happens because of this particular, whichever chemical they receive, the interleukin or the CSF. Oops. So we'll watch that one.
Our body is constantly making new blood cells. Billions of white blood cells, red blood cells, and platelets are produced every day to replace older cells that die, either during normal processes or because of illness. This process by which new blood cells are created is called hematopoiesis. Blood cells in the body, including the white cells, are made in the bone marrow, which is the soft, spongy material in the center of the bones. Blood cells are created from a group of master cells called stem cells, which also live within the bone marrow. These stem cells have the ability to divide and produce all the different types of blood cells. The white blood cells are an important part of our body's immune system. Granulocytes are a special group of white blood cells that play an extremely important role in protecting the body against infections. Neutrophils are a special class of granulocytes that are able to chase, engulf, and digest the bacteria that can cause infections. Neutrophil development in the bone marrow takes approximately 10 days. Mature neutrophils are released into the blood where they survive for only 3 to 6 hours. Because mature neutrophils are so short-lived, the bone marrow constantly produces large numbers of neutrophils to meet the body's needs. Our body makes natural proteins that can signal the stem cell to start making cells that will eventually develop as neutrophils. One of the important proteins in this process is called granulocyte colony stimulating factor, or GCSF. The GCSF protein can work on the cells in the bone marrow that eventually become neutrophils and signal that they should start becoming neutrophils. The entire process by which stem cells develop into mature neutrophils can take about 10 days and includes 2 phases. During the first phase, the stem cells and their offspring can divide as they develop and mature. This step takes about 6 days. Then, over another 4 to 5 days, the cells mature further. However, the cells can no longer divide. The mature neutrophils are finally released into the blood, where they can fight bacteria and protect the body from infection.
So you saw kind of that first phase, they differentiated into the granulocytes, but they still could become any one of those granulocytes. Notice the nucleus wasn't specific. You didn't have any particular granules yet. And then it kept dividing, and then it became very specific, and so you wouldn't be able to go back and become any other type of cell. Oops, I wanted that. So did anybody catch how long does it take for those neutrophils to develop?
10 days.
Yeah. So neutrophils are our most common self-fighting, like the generic infections, right? So we're more likely to get a generic infection than a very specific one. And so leukocytes are going to be part of our secondary immune system, and they're just going to try to attack everything. So we have more of them than anything else. And you saw it takes over a week for that to show up. And that kind of goes along with a sickness you have. You might have noticed a tickle in your throat, a little soreness in your throat for a few days, and it wasn't for maybe a week before you really start feeling sick. And that's because that's when those numbers are starting to really fight the infection, and the winning is what causes our symptoms, if you will. And then how long were they in the bloodstream? Did you catch that? Not long. Hours. They 3 to 6 hours. That is amazing to me that these white blood cells that are so important last not but a few hours each. So that's why leukopoiesis is going to be happening all the time, because they have such a short lifespan. Thrombopoiesis, like the others, is the formation of, the first part tells you what it's making. So in this case, it's making thrombocytes. So again, you have your progenitor cell, and you can see it's differentiating. At this point, it could become a red cell, or it could become a platelet. So depending on signals that it receives, if it receives thrombopoietin, TPO, it's going to cause the cell to differentiate into the megakaryocyte that creates platelets. So thrombopoietin, TPO, we want to know that. We want to know megakaryocyte is the precursor, comes before. This is the precursor of the platelet. The word mega is many or large. Karyocyte is body. So you can see this cell has many nuclei. It's going through division, like cellular division, just the mitosis, the division of the nucleus, sorry, several times. So it has many nuclei, and then it doesn't split the cell itself. So it's a megakaryocyte. And then the platelets are actually extensions of that megakaryocyte that pinch off. So they're not a cell, they're like cytoplasmic extensions. So in this video, you're going to see how that thrombopoiesis happens, and maybe pay attention to how this cell is too big to go through the capillary pores of your blood vessels, right? So it's the pinching, trying to squeeze through, that causes the fractioning of this cell. Oops, well, that'll work too.
Or 15 times larger than most red blood cells. Platelets are small anucleate cell fragments found in the bloodstream. They release a number of growth factors and cytokines and play an essential role in hemostasis. Platelets are derived from megakaryocytes, which are found in bone marrow alongside leukocytes and erythrocytes. The average megakaryocyte is 50 to 100 micrometers in diameter, or 15 times larger than most red blood cells. Megakaryocytes are therefore too large to enter the sinusoidal vessels. Instead, cytoplasmic processes of the megakaryocyte penetrate the endothelial pores. The sheer force of blood flow then stretches these processes, which eventually break off to form platelets and barbell-shaped protoplatelets. The protoplatelets further differentiate into platelets by twisting and pinching along the midline. Platelets remain in the circulation for 5 to 9 days. Any platelets that are not utilized for thrombus formation are eventually destroyed via phagocytosis in the spleen or liver. After producing an average of 1,000 to 3,000 platelets, the megakaryocyte is now small enough to enter the bloodstream, where it is subsequently consumed by alveolar macrophages.
Okay, so it talks about hemostasis, the formation of blood clots. That's what tonight's reading is about. And then you saw hopefully the pinching off of the cells. You guys, our bodies are amazing. Like all the things that happen at the cellular level, even though I can explain like the genetics behind it and how it happens, I just can't fathom like, how does our body know to do these things? It's just so crazy. Our bodies are so smart. Okay, did anybody catch how long platelets last in your bloodstream?
I think it said 5 to 10 days.
Yeah, good. So about a week they'll last. And this is another one of those, like if there's a cut, your injured cells will release chemicals that attract platelets, which will release other chemicals that attract other platelets. So that's all part of the blood clotting process. Okay, so we're down to the last slide set in our notebook. Oh, hang on, I'm trying to go forward a whole slide. There you go. We're on the last slide of our notebook. So you have this to fill in. I gave you writing boxes for each of those. Remember, look at the shape of the nucleus in order to determine which cell they are. And then there are the follow-up questions on the next slide, and I will get to that. How many are there? I feel like there's 8 questions. 6 questions to follow. So you guys are working on this slide right now in your notebook. I'll give you a few minutes to get your answers in before we start calling on people and looking them over. I got 5 minutes on the clock. I'll check on you and see how you're doing. On the board, I got the flow chart. So the hemocytoblast making lymphoids and myeloids. The lymphoid making the lymphocytes. The myeloid, you have to look at the cell in order to identify the type of cell. And basophil, you can hardly see the nucleus behind the granules. Eosinophil, you got that headphone, old-fashioned headphone or earmuffs. Neutrophil, the nucleus never looks the same. It's always in multiple pieces. It might be 3, 4, 5, 6 pieces. Monocytes have a U-shaped kidney bean, horseshoe, platelets, little fragments, and erythrocytes like the lifesaver. And this one was tough for my last hour, so I'm just going to tell you which cell differentiates into platelets. So I'm talking about the precursor to the platelet, which is the megakaryocyte. So I'll leave that up there for a minute if you need to steal any of those answers. And then I'll pop back onto the agenda. Talking about hematopoiesis, practice, practice, practice. Make a flow chart. I think that's really helpful for practicing. And then talk about each one of the cells in the flow chart. So make that like a concept map and then be able to make connecting terms like the chemicals involved or the location, the function, those sorts of things. So we have a 3 levels reading guide going over hemostasis. Finish your notebook if you have follow-up questions to do on any of those slides. Get that turned in today. You can resubmit from last week's notebook, or you can turn it in as an attachment to today's notebook, either way. Okay, then we have 3 levels reading guide. If you need a little more time on that, we have an hour left of class. You probably have time to accomplish both things. But if you need more time, the reading guide you can turn in at a later date, like tomorrow or Wednesday, but the notebook I need today. Okay, so with that, what questions do you have?